This is section 22, Introduction to the T-Test in Making Sense of Statistics. A lot of times as researchers, we need to determine the statistical significance of the difference between two sample means. Now, previously we've talked about statistical significance and p-values. So let's now look at example 1, which talks about the need for making these comparisons of two sample means. A researcher wants to determine whether there are differences between men and women voters and their attitudes towards welfare. So separate samples of men and women are drawn at random and administered on an attitude scale. The women had a mean of 38 on a scale of 0 to 50, where 50 was the most favorable attitude. Men had a mean of 35. Now, so women had a mean of 38, men had a mean of 35. 50 was the most favorable attitude. In just simply looking at the numbers, we would look at that and go, well, women have a more favorable attitude towards welfare. Well, what we need to determine as researchers is whether there is a statistical difference between these two means, 38 and 35. So what actually accounts, what accounts for the 3-point difference between the mean of 38 and the mean of 35? And so, as we just said, one possible explanation is that the population of women have a more favorable attitude than the population of men, and that the two samples correctly reflect the difference between the two populations. And another possible explanation is raised by the null hypothesis. So we've talked about the null hypothesis in the past two sections, section 20 and section 21. And the null hypothesis states there is no true difference between these two groups, between the men and women, that the observed difference is due to sampling error created by random sampling. So around 100 years ago, William Seeley Gossett, while he was working for Guinness Brewery, Guinness Beer, from 1899 to 1935 was his employment there. He developed a test, which is called the T-test, around 1905 to examine small qualities, small samples of beer in basically quality controls, so in the brewing quality control. Now, his employer, Guinness, would not allow him to publish in academic circles, so he published under the name of student. Thus, the T-test is also known as student's T. Now, what his test did, it actually was developed for the situation described in example 1. And what we're trying to do is test the difference between two sample means to determine statistical significance. As a test of the null hypothesis, the T-test yields a probability that a given null hypothesis is correct. Now, remember in section 21, we talked about probabilities, and when there is a low probability that it is correct, say, as low as 0.055% or less, researchers usually reject the null hypothesis. So remember that. Now, so there are three basic factors which interact with each other in determining the probability level. Now, I want to say also that computation procedures for conducting T-test are really beyond an introductory statistics course and this particular textbook. So what we're doing now is just simply describing what makes the t-test work. And so here are the three basic factors which interact with each other in determining the probability level. So the first basic factor is the larger the sample, the more likely the null hypothesis will be rejected. Now, so, there is also, if we look at this, the larger the samples, the less likely the difference between the two means was created by sampling error. So we've already kind of talked about that. The second basic factor is the larger the observed difference between the two means, the less likely the difference was created by sampling errors. And remember, we talk about random sampling tends to create many small differences. And so when larger differences are obtained, the t-test is more likely to yield a probability low enough to allow rejection of the null hypothesis than when small differences are obtained. And then the third basic factor is the smaller the variance among the participants, the less likely it is that the difference between two means was created by sampling errors, and the more likely the null hypothesis will be rejected. And considering this particular principle, we have to think about if there's a population where there is no variance, and everyone is identical, so everyone looks alike, thinks alike, speaks alike, and acts alike, there is less variance. And so the smaller the variance among the participants, the less likely it is the difference between the two means was created by sampling errors, and the more likely the null hypothesis will be rejected. Now, there are two types of t-test. One is for independent data, and this is also called uncorrelated data, where there's no matching or pairing of the individuals across the two samples. So example one that we just discussed illustrates independent data or uncorrelated data, and that was that the men and women were drawn independently from the two populations without regard to whether anyone matches. So we weren't matching them on age or political affiliation or socioeconomic status or education level. So so no one is matched or paired. All right. And then we have correlated data or dependent data, which means that the individuals across the two samples are matched or paired on some factor. So it could be that they are matched on age, socioeconomic status, education level, numerous other aspects that we could match or pair the individuals across the two samples. So our example number 2 on page 121 looks at what we would talk about as far as dependent data. So it talks about the visual acuity, and it's talking about pairs of same-sex siblings. So 2 brothers or 2 sisters were identified, and so for each pair of siblings a coin was tossed to determine which one received a vitamin supplement and which one received a placebo. So for each participant in the experimental group there was a same-sex sibling in the control group. So these participants were matched based on the fact that they were same-sex siblings. So now the results from study in example 2 then are actually less subject to sampling error than the means from example 1. And in example 1 there was no matching or pairing. In example 2, we have matched the two groups. And really when we're talking about that, the genetics and gender are associated with visual acuity. To that extent, the two groups will be more similar at the beginning of the experiment than the two groups in example 1. So here we go. We say the means that result from example 2 are less subject to sampling error than means that result from example 1. So dependent data is less subject to sampling error than means that result from independent data. So you can see how that actually operates through this sentence. So the t-test for dependent data takes the reduction, the possible reduction of error into account. Now, researchers often mention in their reports whether they have conducted t-tests for independent or for dependent data. This concludes section 22. You will want to go back and read page 120 on the 3 basic factors and read that a little bit more in depth than what I covered in the lecture. Our next section, section 23, will discuss how to interpret the reports of the results of the t-test.